I decided to change my talks. So this first talk will be a brief tutorial on cancer. Just I saw that the tutorial that I saw that was given about chemotaxis seemed to be rather effective. So I decided to do this. Then tomorrow I'll talk about breast cancer and outcome prediction. And on Thursday, I'll give a more colloquium type general talk on moving from physics to biology, just sharing my experiences with you. And that talk will cover 3.5 topics. One of them is on transcription, which is the topic of the lecture that I'm going to skip. So you'll get exposed to what I was going to say anyway. One problem is that, as usual, during the third night when I travel, jet lag catches up with me, so I didn't do that well last night. So if I don't understand your questions and don't answer properly, bear with me and it will slowly come. The last item, from the back you won't be able to read most of the slides. So I strongly encourage you, I cannot force you, but I strongly encourage you to move forward. I don't mind you sitting in the front row reading your email. I'm used to it. Lastly, this is a school. So whenever you have a question, interrupt me. There will be no disaster if I don't finish everything that I planned to do. And if you don't understand, you're stuck, don't be shy, raise your hand, get a microphone, and ask a question. OK, so the way first of all, about cancer. Well, people were telling you, Michele was telling you that cancer is passé, we are now into Alzheimer's. Well, we all have to die. And we basically die of 3 things, of heart failure, cancer, and dementia. Now pick your choice. So cancer was the major killer, and heart disease was even stronger killer. Heart disease has gone down significantly, mainly because of, OK, I hope there are no cardiologists here. Heart disease is a simple disease. There is a pump. There is a problem with the pump. You call a plumber. He fixes it, basically. But the biggest advances in in avoiding death from heart failure were prevention. And that is more chemistry. It's complicated. Cholesterol. People take all these Lipitor and all these statins. And that keeps cholesterol in check, and that reduces heart attacks and heart failure significantly. And also, people that don't feel that something is wrong, they are catheterized. People walk into their heart, look, and put in something to keep the arteries open. So all this is preventive medicine, and this has the biggest impact on death from heart failure. In cancer, we are getting there. So there is we know enough about the disease now that we know how to take preventive measures. Let me demonstrate that here. What you see here is the cancer death rates in the US measured from 1930. Actually, I have a plot going to 2008, but only for males. And so I put up this one, which has males on this side and females on this side. So and what you see is the different colors are different kinds of cancer classified by the organ which is affected. And the most prominent, dominant feature in these curves is this one, which from the back you can probably not see, but it's a faint gray curve which shows the death rate from lung cancer. Among American males, this was by far the largest killer. It started to go up in 1930, 1935, 1940. That's when it sort of started to pick up a positive derivative. You can guess why? In the early 30s. Okay, a few years before that, there was the First World War. American males went to Europe and started to smoke. 20 years later, 15 years later, they started to die of lung cancer. And you cannot, I mean, it's really very, very clear. Somewhere around here, people realize that lung cancer, that smoking is bad for you, and they put on the cigarettes all these notes saying that you can smoke, but you will die from it. And then people started to stop smoking, and you see a few years later, the death rate from lung cancer is going down significantly. By the way, American women started smoking after the Second World War, and started dying 20 years later, and they stopped smoking. They didn't stop it. It's an interesting thing that this flattened out. So some people will continue smoking no matter what. Intelligence is not given out evenly. So this, to me, is the simplest way of prevention. You realize that this is bad for you, you stop doing it. I mean, people used to go to the beaches and get baked in the sun, and the blacker you are, the better it is. Now people realize that the skin doesn't like that, so people do it less, and this will probably knock down the melanoma, skin cancer. Okay, so I'm done with this. The disease that we will talk about in the next lecture is breast cancer. It's this one, which was for a while the #1 killer among American women. Then it was overtaken by lung. And it is a pretty bad disease. So the way I set this up is different from the previous speaker's review, in that I'm not giving you a historical review. I basically took one paper by Hanan and Weinberg. They published a paper in 2000. It was called The Hallmarks of Cancer. I recommend to you to read this paper. It's a superb paper. Then a second version came out in 2011, which is more elaborate, has more detail, and less readable. So I suggest that you read the first one, and if you feel like this is an interesting topic you want to get into, read the second one. And what it does, it describes cancer in an engineering way. It says what has to happen for a cell to become cancerous. Cancer cells, the first and #1 property of cancer is growth, abnormal growth. So normal cells have all kinds of regulatory mechanisms that protect against abnormal growth and other kind of disasters. And these regulatory networks somehow have to break down in cancer. So let's go one by one through these regulatory networks or families of networks. And these are the hallmarks of cancer. These are the 4. There are 4 plus 2. I'll explain what this means, is 4 plus 2. There are 6 hallmarks of cancer, which are discussed in these papers. So, okay. So this is a nice diagram, a wiring diagram of a cell. It looks complicated, but in reality, this is much more complicated. This is a caricature which is still readable. So what do you see here? You see all kinds of genes, receptors, effectors, transcription factors that interact and act on each other, and they actually form several categories of networks. This group here, all these circuits have to do with cell proliferation. Cell proliferation starts by some signal which is coming from the outside usually, which tells the cell start to divide. This signal is a chemical in the form of growth factors. Small ligands attach to receptors which are on the cell surface or inside the cell. There are some of those as well. And all kinds of chemical changes take place. Some of these were described by the previous speaker in yesterday's lecture, and even today, I think some of the receptors came up. And these receptors trigger a cascade of molecular events that culminate in transcription. Transcription of all kinds of machines that are needed for DNA synthesis, and the cell can start synthesize DNA and enter the cell cycle. So this is the first, I'll talk about this in more detail. The first hallmark of cancer is that cells do this without proper signaling. Usually it's done in response to some external signaling, and when the cell does it autonomously, that means that this proliferation machinery is broken down. That's the first hallmark. Another one is losing the response to signals for growth arrest. There are situations when a cell gets signals, internal and external, which tell it, don't divide. Cancer cells learn to ignore these signals. Then there are a whole lot of networks that have to do with cell death and viability. I'll talk about all this in a moment in more detail, that have to break down in cancer. And motility, there are all kinds of signals that tell a cell to start moving, which are abnormal. And that gives rise to metastasis. So let's look at now what I'll do in the next half hour, maybe even more, is go through each and every one of these hallmarks, one by one. And what I'm doing is whenever I personally was involved in a study that touches one of these hallmarks, I'll tell you a bit about it. So this way I differ from the introduction of the previous speaker. Not that I think that my work is more important than the very important discoveries that were made by the people who originally discovered these things. But just to show you, to give you some sort of feel that some of these questions can be worked on in some sense by a physicist. Okay, so that's the aim, is instead to try to give the introduction to sort of highlight or very briefly work of my own group on the subjects that we go through. Some of it, not all of it. Okay, so these are the hallmarks. First of all, self-sufficiency in growth signals, ignoring anti-growth signals, evade apoptosis, which is a way for cells to commit suicide, immortalization, I'll explain what that means. Basically, it means that a cancer cell can divide an unlimited number of times. And these are needed for a tumor to sustain itself and be viable. Now, there are two more, angiogenesis. This is growth of blood vessels. This is needed for this tumor to become larger than the critical size, which is very, very small. And finally, metastasis. A tumor can live without metastasis, but metastasis is usually necessary to kill the organism. When a person has colon cancer, he's operated on, the resection, they remove part of his colon, stick it together, he can go on living for as long as he would live without cancer. The way colon cancer kills is by metastasizing to the liver, to the lung, and to the brain. Okay, you cannot live without the lung, you cannot live without the liver. Some people can live without a brain, but it's not a good life. So these are the 6 hallmarks of cancer, and I'll go through them one by one. Here are again a figure that describes them, the 6 hallmarks. So I will start with sustaining proliferative signaling. So I guess you know about the cell cycle. And I'll just briefly say that there is a phase, which is a gap phase, where the cell sits there and prepares the machinery to start synthesizing DNA. It has to pass a checkpoint, and then it goes into the synthesis phase. When DNA is synthesized, DNA is replicated, so the new strand has to be synthesized. Then it stops, it goes into G2, another gap phase, where it again has time to prepare material and energy in order to go to the next phase, which is mitosis, when the two strands of DNA, the one that were just copied, are separated into the nuclei, and the nuclei of the two daughter cells become two distinct nuclei, and the cell splits in two. So that's the mitotic process. And after mitosis, the cell again goes into this G1, where it has to grow in the sense of becoming bigger, accumulate biomaterial, and then it can do it again. And there are checkpoints at the entries. Here there is a checkpoint to get into the S phase. There is a checkpoint to get into the mitotic state. So there is a lot of checking going on for a cell to see that everything is okay, that yes, proceed, do it. And if something is wrong, or if some process has to take place which cannot coexist with cell division, then the cell goes into a phase which is called G0, cell cycle arrest. No more divisions until a signal comes which kicks it in from G0 into G1, and it goes again. So that's reversible. There is terminal differentiation that is not reversible when a cell reaches a state where it doesn't have to divide anymore and shouldn't divide anymore. Most of the neurons are in this terminal differentiated state. The liver cells are apparently in this state. They can, if the liver is damaged, then the liver cells can start dividing until the liver again attains its normal size. And there is senescence. Senescence is something I'll explain in a while. Okay? So these are the circuits of sustained proliferative signaling. And I told you that this proliferative signaling starts with some growth factors. This is a caricature. It's poorly done, but it's clear. So this is the cell membrane. Here is the outside of the cell. Over there is the inside. This is the membrane of the nucleus. The nucleus is here. So the growth factor is represented by this thing here. And this is a receptor, a caricature of a receptor, of a cell membrane receptor. And let me show you a caricature of what happens. So the growth factor is attached to the receptor, and in response, the receptor undergoes a conformation change, or some chemical modification, like phosphorylation. As a result of that, this molecule, which is called an effector, gets modified, changes its shape. And when it changes its shape, it is released from the receptor to which it was bound. It attaches to some other protein which is in there. The two form a complex which is active. And in this case, it is an active transcription factor. It enters the nucleus and attaches to the promoter of a gene, and transcription can start. So this is a very simple caricature of what's going on, of a much more complex reality. So let me show you this complex reality in a movie, okay, which gives you a slightly more faithful, what does it want? What do I have to do? These things. Yes? I couldn't hear you, so I cannot follow your advice. Why when one of you comes here and shows me what to do? It's easier. Let me see. Let me try again. Yeah? Please. Oh, this I should. And nothing happens. So try it again. Okay, good. Thank you. Okay, so maybe I could restart it. Or maybe I shouldn't experiment with it since it works. So these are cells. And what you will see is that this is a receptor tyrosine kinase called EGF receptor. And when the ligand comes, the receptors dimerize. Two of them hug the ligand, and their legs, which are inside the cell, act on each other. They phosphorylate each other, and that starts the entire cascade of events that hopefully now you will see. Okay, so we now focus into this. This is the cell, the nucleus, and we go to the surface, and the EGF molecules are swimming around. Here is a receptor, and it's bound. It changes its conformation. It dimerizes with another one. The dimerization brought into physical contact the two legs. They phosphorylate each other, and now all kind of stuff happens. Things bind, get modified, phosphorylated, and activated RAF is binding with MEK, and the transcription factor will be resulting out of this ERK, and then it will go into the nucleus and start transcribing all kinds of genes, which will do, for example, if the gene that is transcribed is DNA polymerase, then you can start synthesizing DNA. RNA polymerase, you can start synthesizing RNA. So this is a very nice picture. Mine is somewhat simpler, the one I showed you. Okay, so we went through this, so let's do it quickly. And then the question is, okay, the last, okay, just a sec. And the last thing that I didn't mention is this. This process starts. Transcription goes. Something has to stop it. And there is a whole machinery built in of negative feedback which takes this process which was started and stops it. And there are several levels at which this acts. Okay, so now the question is, what can go wrong? Because we said this is what a normal cell does. In a cancer cell, things break down. And there is more than one, one way you can mess up the brakes of a car. Neighboring tissue of the tumor to send growth factors to it. Which is an interesting thing, by the way, because it goes in the direction that cancer research is going. It doesn't disconnect the tumor from its environment, from bacteria that live in it and near it, from neighboring cells, the reactive stroma. All these should be thought of as one unit that produces the disease. So in this case, many times, it's the neighboring healthy tissue which produces the growth factors that make the cancer grow. Okay, what else can go wrong? So this is the signal. The signal can be stronger. Everything is stochastic. You understand that. There is so much stuff floating around, so many receptors. Receptors can bind so many things. So it's a single ligand which hits a cell is not enough to trigger growth. So there must be a critical concentration of the ligand, and that's why this will help. Another thing is, of course, what you can do is to increase the number of antennas that receive the signal. Then you will be sensitive to weaker signals. And so growth factor receptor amplification is a major manner in which cancer cells do this thing. They just make many copies of the DNA of this receptor, which get transcribed, and therefore you have more protein, receptor protein, than normally needed. Okay, the next thing that can go wrong is that the receptor is mutated. We said it goes into this active state as a result of the binding of the growth factor. Now it could, because of a mutation, be activated irrespective of growth factor. Okay? And then it will activate the effector, and everything will go irrespective of growth factor, independently of growth factors. Okay, the effector can be mutated. If the effector has this shape to begin with, then it doesn't need the signal to be active. Then it's called an oncogene. And the major oncogene is RAS. It's a hub. It plays a major role, and when it's mutated in many cancers and causes proliferation to go. Finally, the negative feedback mechanisms can be impaired. This whole thing starts and doesn't stop. Yeah.
If it is independent on growth factor, then why it is needed to be increase the number of growth factor?
It doesn't need to. I asked what can go wrong. Any one of these steps will produce autonomous growth. Any one. You don't need more than one. One is enough. Okay? Thanks for the question clarifying this. Is it clear? So there's different things that can go wrong. Not all of them have to go wrong. Okay, so cells see a lot of signals and respond in different ways. So this is a study that we went on some years ago, and there were, oops, I will break my leg at the end with this. Okay, it was a group in collaboration with Yossi Yarden, which has been ongoing, and we are still collaborating. And this was one of the first major ones. She was my PhD student. I show you on every study the PhD student who worked with me. I don't show the students of the other guy. Okay. So what we did, let's try to understand the transcriptional, time-dependent transcriptional profile of a cell that is undergoing this process. So what we did, we measured as a function of time the level of expression of the full genome of a particular cell line which was stimulated by EGF. Okay? And watched what goes on, what goes up, what goes down. And these are different phases of the response. First of all, at time 0, the EGF is administered, and right away, within a minute, the receptor is phosphorylated. That is a post-translational modification. We don't see that in the mRNA data. You know about transcription, translation, all this stuff. So this step is not seen. It's fast. Then what happens next is the activation of the effector. This is usually also post-translational by phosphorylation or by just a conformational change of the protein. Now, transcription starts at around half an hour, 20 minutes, and then we should see some signal of some transcripts going up. What we measure is mRNA. Then, after an hour, proteins are already produced in response to this signal. That's the time scale. And after a day or half a day, you already see a phenotypic response, which can be, in this case, either cell division or the cells start to move, depending on what kind of cells and what kind of stimulus you get. Okay. So in this case, what you see, you give EGF at time 0, and you look 10 hours later, and this is a cell culture. And what is dark is biomass, is cells. And what you see that the amount of cells has gone up, which means that these cells divided in response to administering EGF. And this is the result of our analysis of the expression data. So we took out of the 20,000 genes, we do it with a microarray. Have you been exposed to that technology? It's a device which measures the expression levels of mRNA transcripts for 20,000 genes. So you get from an experiment, a single experiment, 20,000 numbers. And we've selected out of those 400 that passed some filter. The filter being that they went up. the level changed with respect to time zero. The reference is what happened at time zero. Time zero is when we give the cells EGF. And we compare the level of expression sometime later to time zero. And the fold change of expression is plotted here. So what you see here is every column is a time point. To zero is when we give EGF and start measuring. 20 minutes, 40 minutes, 1 hour, 2 hours, and so on. The time, every column is a time. And every row is a transcript or a gene. And the color that you see represents the level of expression of that gene relative to its level at time zero. Red means very high, blue means low, or the same as time zero. And so when you look at 20,000 genes and you ask how many pass a filter being that at its maximal level of expression, it is at least 2 or 3-fold higher than at time zero. And you find that 400 genes do satisfy that. So these are the 400 genes. Now, they are not arranged at random. Yeah. Oops. This is not going to work. Yeah. Okay.
What would happen if you just took the cells at time zero, you didn't administer anything,
and you let it be?
And then you measured, I'm just trying to ask how many false positive would you get? Then if you measured it at 20 minutes,
well,
how you would just go up and be a 2-fold change just by, I don't know, just by...
We didn't do that experiment. And my guess would be that it would be a very small number. There is noise in the data. And we control noise. I would rather not go into detail, but we are very careful about noise. We estimate the noise. Usually in experiments that we do several repeats of the same experiment, and then we just estimate the noise, and we know what is the probability of getting onto this list by random fluctuations, okay? And we try to keep that probability below 5% or something like that, and that sets the value of the fold change that I'm showing you. So I cannot say that these are all real, but I would say less than 5% are false, okay? So, these genes are not at random. You see some structure here. This is because we arrange them like that. We group them together to the maximum value of their expression. And all these have their maximum value at 40 minutes. All these have their maximum at 60, 120, and so on. And you see a wave of activity running through the system. Okay, so we looked at who are these genes, and now we focused on those that have transcriptional roles. And we were amazed to see that a small fraction of them, colored in green, are transcriptional activators. And the large fraction of them are actually suppressors. About half of the genes that go up in response to EGF, don't worry. About half of the genes that go up in response to EGF are involved in breaking the system, in the control. So you understand the weight that these cells give to controlling the growth process. So they realize that this is something very important. It's very important to make sure it doesn't go out of hand. Okay. So these receptors are actually involved in many cancers and in a large fraction of the sick people. And therefore, they are a natural target for drugs. And I just listed a few of the drugs that act on different aspects of the functioning of these receptors. So some of these names you may have read in the newspaper. They are the drugs that people use against many forms of cancer. Okay, so this concludes the first. Are there any questions about this? About autonomous generation of growth signals? Okay. So we go to evading growth suppressors. So most of the growth suppressing signals are channeled through this protein here, which is called RB. RB is called the retinoblastoma protein. It was discovered following work that a physician did on children who have tumors in their eyes, in their retina. And it was, the name of the person is Knudsen, and he realized that there must be a two-hit mechanism here, that there are two copies of this, of something. There was no, this was in the early 60s, no genes, two copies, and there must be two hits in order for this cancer to happen in some of the patients, some of the children. Okay? And then Weinberg identified the gene and sequenced it, and that it's called RB, retinoblastoma protein. So what does this particular protein do? Well, it arrests cell cycle by binding to something called E2F. E2F is a family of transcription factors. And what they do, they transcriptionally activate a whole bunch of stuff needed for DNA synthesis. DNA polymerase itself is one of them, cyclin E, and several other components that are needed for the cell to synthesize DNA. E2F is elongation factor 2. It's a family of transcription factors. So what does RB do to it? It binds to it. And bound, this complex, RB-E2F, cannot act as a transcription factor, and it's inactive. And therefore, DNA synthesis will not take place. What happens when you want to enter the cell cycle? So when you want to enter the cell cycle, you have to somehow break up this RB-E2F complex. And the way that is done is by phosphorylating RB. When RB is phosphorylated, it changes its conformation. It does not bind anymore to E2F. E2F is released, and it can go. So now the way biologists draw this, if the process you want to achieve is this, E2F activates the process. RB inhibits E2F. This T-shaped thing is inhibition. Arrow is activation. So when RB is phosphorylated, E2F is released, and the whole thing can start. And this is done by a complex of cyclin D and cyclin-dependent kinase 2, which is a kinase, and RB is one of its substrates. Okay. Now, if cyclin D is present and you still don't want the cell growth to take place, there is a break called P16. So if a cell decides it doesn't want to grow, it transcribes and translates and produces P16, which then inhibits this complex. I cannot see red. Sorry about that. It says P16 here. So this holds the thing in check. And this is one of the main processes that cells use to control and provide anti-growth signals through the RB protein. So again, what can go wrong? Many things can go wrong. You can hit this path anywhere. The main problem which one has in retinoblastoma is that RB itself is mutated. So it's mutated, and then it cannot be phosphorylated, and then it's active, and E2F is active all the time, and the tumor goes. Okay? Okay, there should be a number 3 there. This is the third hallmark, evading cell death and apoptosis. Now, apoptosis is an amazing behavior. It's the most altruistic behavior you can think of. Here is a single cell, and it senses that things have gone out of hand. There is such damage that it cannot possibly be repaired. So what it does, it commits suicide because it understands, because if it doesn't, then that will endanger the life of the organism. So it's, you understand that how this thing evolves. Organisms that pick this idea up have an advantage over organisms that don't. And therefore, apoptosis is a very sensible way to go for the organism. P53 is a very conserved protein. And it's right there. So P53 is a central player. It is the guardian and the detector and the handler of all kinds of stresses. 50% of human tumors, roughly speaking, have mutated P53, a loss of function mutation. P53 is unable to perform its function of inducing apoptosis. Was this too fast, or is the pace okay? Anyone has any questions, please? Okay. So let's look at P53, and this is a study we did many years ago. I show it to you because I like it, because it was one of the first things I did when I got into biology. And it was done, the work was done by a very, very nice guy, an Indian postdoc in the lab of Givol, who was a senior biologist that collaborated with me on this. And he was my student who really led this research. So there is P53. It's a protein which is really of central importance. Half the Weizmann Institute, where I come from, roughly works on P53. So it really is a big thing. I'm overstating, but not half, but let's say 30%. And one of the people who there, Moshe Oren, who was the first one to clone P53, came up with a mutant, discovered the mutant, which is temperature sensitive. So at 37 degrees, the cells live happily, and the conformation of this protein, P53, is abnormal. It cannot do its function. You cool the system down to 32 degrees, P53 clicks into the native state. Okay? So just to show you that this is indeed the case, here is an experiment. These cells, it's the same cell line. You knock out P53, or you have, when you put, and here you put in that temperature-sensitive P53. For a physicist, it's easy to say knock out, put in. I mean, each of these steps take ages. And then, so you have the same cells without P53 and with this mutant. You put them at high temperature. P53 is in its mutant conformation. Okay, and you see that there is quite a lot of cells, which means, okay, sorry, and you hit these cells with two oncogenes. These two oncogenes, normally, if you give them to cells, they grow, as they do here. High temperature, no P53, they grow. Low temperature, with the P53, no growth. The cell die because they feel that something is wrong. There is active P53 here. So we use this system. How do we use it? We use it as a switch. The aim of the exercise, I didn't say that, was to determine the primary targets of P53. As I showed you, P53 is involved in a whole lot of processes, and its main activity is transcriptional. So the question that we asked, who are the targets of P53? Some were known, and there were probably out there quite a few which were not known, and we wanted to find them. That's the aim. So we took a switch, this temperature-dependent mutant, and we lowered the temperature to 32 degrees fast, and that's our time 0. Now we have P53 in the system. The cells are under stress, so P53 will start transcribing all kinds of targets. So what do we expect? We expect to see that a whole bunch of genes go up. So these are the targets of P53, right? Well, not really, because some of these will be primary targets of P53, real targets. P53 binds to the promoter, then is transcription of these genes, and protein is produced. But sometimes the protein that is produced is itself a transcription factor which activates some other guys. And they will also go up in response to P53. So what we want to identify are the primary targets of P53, those that really P53 activates and not the son of P53. So how do we go about that? We use a compound called cyclohexamide, which is an antibiotic. It binds to the ribosome. Basically, it blocks the hole in the ribosome. It blocks translation. So transcription goes on as if nothing happened, but the transcribed RNA cannot be translated into protein. So the primary targets of P53 go up. The mRNA goes up. But they are not turned into protein, and therefore they cannot activate the secondary targets. Simple, right? Is there a question about this? Yeah, please take a microphone before I try to approach you and crash.
What's the time scale of these experiments? Because cyclohexamide itself can kill the cells. I mean,
absolutely. So you see too far. So here it says, this is hours. Time 0 is when we lower the temperature and either do or do not add cyclohexamide. And then we measure the response of the mRNA at 2, 6, 12, 24 hours. And they are able to live with, they are able to live with cyclohexamide for a day. Okay, my doing this is done on purpose to keep you guys awake. So the time scale is 1 day this is the experiment. I should have been more careful. This is the experiment without cyclohexamide. With cyclohexamide, we stop after 12 hours. At 12 hours, they already start to be unhealthy and dying, okay? So that's why the cyclohexamide experiment has more time points, but less time. Good point, okay? More questions? Yes, please.
What if the low temperature is activating a altogether complete different pathway and not the actual pathway that happens in the cancer?
Say it again. You ask about the effect of the temperature?
Yes.
That's why, I mean, biologists are good about these things. If you see, this experiment was done also on the control with no P53 and here to show that the temperature has very little, if any, effect on the system. Just the temperature. Here, you see this from where you sit? So this is a Petri dish with cells. The black spots are cell colonies. And what you see here and what you see here is fairly similar. This is at 37 degrees, this is at 32 degrees. So, but just by stepping the temperature, we didn't do much change in terms of the process that we are looking at. So you can go back and ask, maybe there is some process ongoing which is temperature sensitive and is not reflected in proliferation. That will not be seen in this experiment, okay? Or death. So whatever is not involved in death or proliferation and has a temperature effect will go under our radar. Yes?
There might be some other genes also, right? Those are not targeted
by P53.
P53. So what happens to them when you add cyclohexamide?
Okay, good question. Hang on, ask me again after the next slide, okay? If it's still there. At the end, shout. Okay, I think I heard you, and I think I know what the question is. Let's go back after the next slide, okay? Same there, just let me get to the next slide. So this is what I expected, okay? So without cyclohexamide, there is a whole bunch of genes that go up. Some of them are primary targets, and some of them are secondary, tertiary targets. I put in cyclohexamide, only the primary targets will go up. So this is what I expected, that there will be a subgroup of those that went up without cyclohexamide that will still go up with cyclohexamide. So, okay, so this is a physicist's view of the world, because biology is, of course, much more complicated, and what really happens is that. So here are the genes that go up without cyclohexamide, and indeed only a subset of them goes up when cyclohexamide is added. But look at these. There are 90 genes that are upregulated by adding cyclohexamide. So what's going on? Well, what's going on is that p53 activates so many targets. Some of them are transcriptional suppressors. So if p53 activates someone called X, and X is a suppressor of someone called Y, then Y will go up if I block the translation process. Okay, so cyclohexamide has positive targets, which are actually the removal of negative of suppressors. Think about it for 20 seconds and then ask questions if you have any. Questions? Is it clear what I tried to explain? All right. So now we ended up with 38 targets, so candidate targets, and then we extended them. The way we extended them was by looking at, we had 250 or 60 or so genes that went up, and what you see here is again a heat map. Every column, first of all, these 4 columns are the experiment without cyclohexamide. These are the experiment with cyclohexamide, okay? And these are the time points, 2 hours, 6 hours, 12 hours, 24 hours in the first case, and 2, 4, 6, 8, and 12 in the second case. So we have 2 time series, and the colors again represent fold change with respect to time 0. So what do? You do clustering. Clustering means is a name for taking things that are similar and pulling them together. And we invented a clustering algorithm in the mid-90s called superparamagnetic clustering, which uses the physics of disordered ferromagnets to do clustering. So it's a case where you use a physical model to solve a problem in data analysis, basically. And then you use it in biology. And what we pick up in clustering, we pick up groups of genes or transcripts that have similar profiles in the 2 experiments. And we look, these red X's, these are the genes, right? And the red X's denote those 38 candidates that came up as going up with and without cyclohexamide. So these were our candidate targets for cyclohexamide, and we used them as sort of the bait to fish out other genes that behave like them, that for some reason didn't quite pass our filters. The reason for this, I didn't explain that, I'm sorry. Why are we doing this? In order to get these genes, we used all kinds of filters. To get on this table, on this list, a gene had to go up at least 2.5-fold at least 3 time points. It was a very high sort of bar. And whenever you set bars in an experiment like this, you have to play with it and check how sensitive your results are to these arbitrary selections of thresholds. And that's what we did here. So we went in and we looked, is there some gene who looks very much like these targets and should be together with it, but it didn't quite make it in one of the 2 experiments to get on the list. And then we also looked at the detailed profiles. So for example, these are all these putative targets are in this cluster, in this group of genes. And inside it, there is a subgroup A, which doesn't have any one of them. So we looked at its profile, and its profile was indeed different. So all those that were there, we threw out. So this process of enlarging and pruning returned, instead of 36, 38, we had now 47 targets that go up. Okay? So this was the kind of analysis we did. In another step, we released enormously the thresholds. Now we have 1,000 genes to play with, used, again, the same method, ended up with, again, a list of targets, of candidate targets from this very, very large set. And it had, and 75% of those that we found before were also in this list. So we are fairly confident that those 40-some that we identified are indeed primary targets of P53. And indeed they are. This paper is widely used. And we did the same thing afterwards with the genes that go down in response to P53. Immortalization. That's the 4th hallmark. Yeah. Yeah. Last column. Here? Here. Well, you can see that this is the average profile of a group. So individual genes can go up and can fluctuate. That gives you also an idea of the noise in the system. Usually you expect a monotonic behavior to some extent. You have to go up and then it can come down. But you don't expect fluctuations like this on a scale of hours. So there is some noise in the system. Okay, so 3 hallmarks we went through, right? The sustained growth, ignoring anti-growth signals, evading apoptosis, which is by introducing a defect in P53 mostly. Number 4, immortalization. Okay, so this goes back to the early 60s, an experiment by Hayflick, which is a very basic experiment in biology. And very simple. He took cells in serum and grew them. And they grew happily and divided happily. And he measured the time between consecutive divisions and plotted the time between consecutive divisions as a function of the number of divisions. Is it clear? I hope it is. And what he saw, that the time interval between 2 consecutive divisions diverges as you go down in the number of divisions. Around 80 divisions, that time interval is infinite, which means that after 80 divisions, cells stop dividing. And this is very reproducible, stable, correct. Which means that the cells have a clock. A cell knows how many times it divided. And how does it know? The answer is that when DNA is replicated, in every division DNA is replicated. At the end of the chromosome, of each chromosome, there is a region which is called telomere. It's a region of DNA which has about 1,000 repeats of a 6-mer, of a sequence of TTAGGG. This is repeated 1,000 times. This is called the telomere. And these things, from the 2 strands of the DNA that form the double helix, okay, they open and they cross each other and bind so that the end of the DNA is not free. It's not like this. And this protects the DNA from being degraded and being attacked, and it is essential for the viability of the cell. When this DNA is replicated, the telomere gets shorter by 50 to 100 nucleotides, right? You can imagine that when the DNA polymerase runs on the DNA, it stops before it reaches the end, and the region where it sits cannot be replicated. So this is a rough mechanical explanation. So it has to become shorter. Okay? So cells cannot divide indefinitely. Cancer cells do. They are not there. They found a way to become immortalized. And this is achieved by expressing an enzyme called telomerase. What telomerase does, whenever the cell divides and the telomere shortens, telomerase comes and extends it to preserve and maintain telomere length. This is the clock, which cancer cells learn to violate. So you can ask, how come other cells don't do this? Well, some cells do. The germline cells do. Sperm and ova have telomerase, but none of the other cells. Telomerase is silenced in all the other cells of a mammal. So this is the fourth hallmark. And there are two more, angiogenesis. It turns out, now this is bad news in some sense, each and every one of us walks around with cancer cells floating around with his body. Potentially cancerous cells. But they don't become cancer. Potential cancer cells, that means that they have acquired all kinds of defects that I talked about. And they start to divide, but they will not get beyond some critical size. And the reason is that in order to divide, the cell has to get nutrients and oxygen. This is supplied by the bloodstream. Now, cancer grows like a balloon. It's like when you inflate a balloon. It goes like this, becomes a mass. There are no blood vessels in there. So oxygen, the diffusion length of oxygen in human tissue is on the order of several hundred microns. So in order to get enough oxygen and nutrients, the cell has to be in proximity to a blood vessel. So when a cancer cell reaches a critical size, it either stops growing and dies eventually, or somehow it causes blood vessels to grow into the tumor. This is called angiogenesis, this process. What you can see here, this is a tumor in a mouse. And you can see the ingrowth of very, very disordered and dense network of capillaries. So this is the fifth hallmark. The sixth is metastasis. As I said, in order to kill, many cancers have to colonize other vital organs. Okay? It's not a trivial process. It's a major undertaking. The tumor grows. That's okay. It has to grow to have a strong enough for a driver to grow and to break through some surrounding tissue, invade, to get through the muscle tissue that's next to it. And then cells from the tumor have to detach. This is very rare. It's very rare that a cell from my earlobe will start wandering and end up in my toe. Cells are attached to their neighbors. They feel comfortable where they are, and they communicate, and they have all these devices that sense that there are neighboring cells of the right kind next to them. They don't just get up and leave. So there must be some hit which allows the cells to acquire this ability of getting up and detaching from the mass to which they belong, from which they were generated. Then the blood, they have to reach the blood vessel. In order to get from the colon to the brain, the traffic is through the blood. So this cancer cell has to get into the bloodstream. Not easy. It has to somehow squeeze in between these endothelial cells that form the blood vessel. And once in there, it migrates. At some point, it has to stop migrating, attach to the cell wall, to the wall of the capillary, and squeeze out. Okay? Each of these processes is non-trivial. And then it has to find a place to colonize. A cell from the epithelium of the colon is very different from a liver cell. So it has to be able to reach the liver and sit there and grow there in that environment. Not simple. So all these properties the cell has to pick up in order for the tumor to become metastatic. Okay? So this is the sixth hallmark. Now, the next step, so let me pause and see if there are any questions about this. I started at 11:15, right? So I've been talking for 1 hour and 10 minutes. So I have 20 minutes to go. Okay, yes.
So what's the rough number of how many of these pre-cancers you have in the body?
I have no idea. I have no idea. Good question. But there are numbers in the literature. I don't know how reliable they are. It's not easy to measure these things. But there are circulating tumor cells in one's bloodstream, and there are precancerous cells. There are estimates, but I don't think that anyone has actually measured this. The question was he's worried how many of these he has to catch. Yes?
What is the typical time scale of flowing of those cells to the bloodstream?
What is the typical time scale for a tumor to metastase?
Yeah.
Well, that is very, very hard to answer. It depends on the tumor, and it depends on also on the kind of cancer. It depends on there is a large variation. Prostate is slow. So they discover prostate cancer in men who are like, I don't know, beyond a certain age, and then they say, live with it, as long as it doesn't bother you too much. Because you will not die of that, and the probability of dying of the operation is higher than metastasis. So anyway, this is not funny. I mean, this is exactly what the kind of calculation that an intelligent physician does. So it's a slow process. With colon cancer, it's also not that fast. With some of the breast cancers, it's fast. When they discover it, it's already spread to the lymph nodes and spread to other organs. In some cases, the tumor is in such a place where it doesn't, there are no effects. You don't know that there is a tumor there, like pancreas. You know, pancreatic cancer, people die within months. And the reason is that when they discover the cancer, they already discovered that there are metastases all over. So there is a wide variation. But the thing is that in order to acquire this capacity, the cells have to pick up all kinds of alterations in addition to all those that I talked before. So that takes some time. And then what is the mutation rate? So in some cancers, the mutation rate is higher than in normal cells. One of the hallmarks of cancer that I didn't describe is that cells, cells have all kinds of machineries that correct errors. Mutations happen when DNA is replicated and there is some mistake. So there are repair mechanisms. And when those fail, then the mutation rate shoots up. And then things are accelerated. So it depends on what is hit in the cancer to answer the question. And it's a very good question, and one of the things that people are wondering about, don't know. If I take out the primary tumor and I analyze it to death, can I predict whether this is going to be metastatic within the next so many years or not? My next talk is on this question, and it's not clear at all. that the signature of the mutant of I'm going to be metastatic is already in the primary tumor. Sometimes it is, sometimes it's not. Okay? So, yeah.
In brains, neurons generally don't divide. Okay, I just want to know about neurons. Neurons generally don't divide. So, what is the probability of getting a brain cancer and cancer in some other parts?
Okay, it's a good question. Apparently, there are some brain neural cells that divide. Like most of the cancers are glioma, so they are with the glia.
Okay.
And apparently, there is some division there. Division and mutations can be caused by all kinds of other damaging agents, like oxidative phosphorylation. There is all kinds of oxidative agents that are there. They can also damage the DNA. And the chances of damaging DNA are much higher when the DNA is open. That's why the dividing cells have a higher probability of being hit. But okay, so what I'm trying to tell you that I don't really know the answer. Okay? But there are mechanisms that non-dividing cells can have mutations, and there are also dividing cells in the brain. So these are the two ways I escape your question, because there are cancers of the brain. Okay, so what I'll go now through until we are done, which is in about 10, 15 minutes, is to look at reasons. Okay, there are two emerging hallmarks. I'll just talk about them briefly. Two emerging hallmarks. This is in the paper from 2011, and that's why they are emerging. One of them is really very important, and it really should be an independent hallmark. We have an immune system. The immune system is recognizing stuff that is not right and destroys it, living stuff. So you can ask, a cancer cell will have all kinds of proteins which are abnormal. How come that all the machinery of the immune system does not recognize that? Some of it must get somehow into the surface of the cell so that the immune system can recognize it and attack that cell and destroy it. So apparently cancer cells found ways of evading the immune system. Not all. So one of the things that one of the modalities of therapy that people use and are very optimistic about is immunotherapy. Is to try to excite the immune system and use it to fight the cancer. Okay? And in my lecture tomorrow, you will see an example of a particular group of breast cancer, which are called basal, that are in fact belong to two different subgroups that differ in the level of involvement of the immune system in these tumors. So some tumors are doing a better job in repelling the immune system, fooling the immune system than others. But all of them have to somehow handle this problem of evading the immune response. Another one is deregulating cellular energetics. Cells, and I'll talk very briefly about this, and then I'll talk a bit at more length about how, what they call enabling characteristics, what generates these alterations. I already said mutations, but there are a few more. So that's where I'm headed. If I don't finish this today, then I'll start tomorrow here and then sort of morph into breast cancer. So the genomic modifications that one talks about are, first of all, point mutations. You heard about this probably from Boris Schreiber, but you know what point mutations are. When the DNA is a text made of 4 letters, TCGA. Okay? So when the DNA is replicated, there may be a mistake, and the G is replaced by a T. And this mistake, you know that every 3 letters, if this mistake happens in a coding region, every 3 letters stand for an amino acid. So in many cases, switching a letter could give rise to a different amino acid. And then the protein is different. And it can lose its function, or it can even gain function and become an oncogene. So these are point mutations. So you understand this. I'm not going to explain this. Translocations are something else, and I'll show you in a moment what that is. There are copy number changes, amplifications of deletions of regions in the DNA. And there can be insertion of viral genes. You know that viruses attack cells, and they incorporate their DNA into the cell's DNA, and when the cell replicates, it also produces the viral proteins, which are oncogenes, cause cancer. I don't have a slide on that, but I'll mention that in a moment, in one of the next ones. Okay, and there are also, these are all genetic alterations. Genetic means that the text on the DNA has been modified. Then there are also epigenetic alterations. These are changes to the DNA which do not involve change of the text. And there are two that I will discuss, methylation and histone modification. So this is where we are headed, okay, for the last 5 minutes. So deregulating the cellular energetics, the Warburg effect. It's a very interesting effect. And let me try to explain it. I'm not an expert in metabolics. So this is the metabolic circuitry of a normal cell. Okay? It takes food, in particular glucose, all kinds of sugars. Let's look at glucose. And it does all kinds of steps to produce ATP. ATP is the molecule which carries energy in a cell. This is how cells, what cells usually do in terms of managing energy, they take some form of energy from the outside, okay, and they do something with it which is orderly, like producing ATP, and the rest gets dissipated. Okay? It's the standard thermodynamic machine, but it's pretty efficient. And so there is some ATP produced at this step, and here, this is called glycolysis, turning glucose into pyruvate. This is a fermentation process. And very little ATP is produced here. The main source of energy is the pyruvate is being transported into the mitochondria, which are really the factory for energy of the cell. And using a lot of oxygen, the pyruvate is decomposed and a lot of ATP is produced. So most of the ATP is produced in this step, which uses oxygen. This is what normal cells do in order to produce energy from food, from nutrients, sugars. Cancer cells mostly adopted this part of the process, glycolysis, which is blown up here. This is that box. And here, no oxygen is used, and only a small amount of ATP is produced. And they prefer this. This is called the Warburg effect. This is Otto Warburg who discovered this. And there are lots of papers that talk about this. And I don't think that they really understand why. Why, in the sense, in biology, why has several meanings. One of them, the deepest one, is what is the evolutionary advantage of this? How does this cancer cell gains some advantage, proliferative advantage, by switching from oxidative metabolism to glycolysis? Now, sometimes it's understood. Some of the cancer cells live in a hypoxic environment, which means they don't get a large amount of oxygen. They are not anoxic. They are not without oxygen. Then they would probably die. But they are not normoxic either. They are sort of a low level of oxygen, but not high. Then they have to, under hypoxic conditions, they have to find some trick to produce energy, and they would go this way because there is no other way. But also cells that do not suffer from lack of oxygen switch to glycolysis. And that is, to me, a puzzle. Yes, you had a question? No? Actually, this chair worked.
Yeah.
Yes.
So, could this be that blood vessels that it needs to grow, the cancer cells, the blood vessels
Yes.
for oxygen? So, in this case, it doesn't need those blood vessels to grow this could be one reason?
Even this needs some oxygen. You cannot, there is no life without oxygen. So there must be some oxygen. So there must be some supply. But in some cases, that supply is meager. So what you are saying makes sense. It says, I'm secreting VEGF and all these factors which attract the blood vessels to grow towards me.
Yeah.
And I'm doing my best and no blood vessels came. I have to survive somehow, so I switched to glycolysis, which is more or less giving a better explanation to what I said, because I said I went already to the end point. I have some oxygen, not as much as I would like to have, but some oxygen. I have to make do, and this is what I do. So your question asked gives the reason for that, how you get there. But that's the mechanism that you mentioned. Yeah.
Could it also be that for some of the cancers, maybe you've screwed up the DNA repair mechanisms, which means that then if you decouple the mitochondria, you're going to get less DNA damage, so you don't need to repair that much. Meaning if you had coupled in the mitochondria also...
Look, you're a physicist.
Yes.
It's a nice model. Why don't you now spend the next 3 years doing an experiment testing it? Okay, it could be. I have never seen an experiment that showed that by going this way, you reduce the damage, the DNA damage, and gain that in that fashion. Could be. It's an interesting idea. I've never seen it in print.
Okay.
But that can mean that it's great, or it can be that it's wrong. But in biology, if you have a good idea, then you go to a lab and figure it and try to show that it actually is. Yes.
Is this common for all kind of cancer?
Nothing is common for all kinds of cancer.
Okay, so is it
even within the same tumor, some part of it will do one thing, and some part there will be heterogeneity on all scales. But it is very widespread. Not all cancer cells switched to glycolysis, but a very large fraction of cancer, of types of cancer and cancer cells. Yeah.
So if this is the case, do we know like how many general cells which are not cancerous do this thing?
How many?
Like normal cell. There might be some cases which switch
not that I know.
Okay.
Not that I know. We don't ferment.
I mean, generally, but yeah,
I mean, there are all kinds of tricks that cells use when they are under anoxic conditions, you know, all kind of anaerobic and aerobic. And so under stress, if there is oxygen, there is stress, there is no oxygen. If people exercise and they run, and then there is a switch. Maybe you are right. I think you are right. So all these marathon runners and so on, at some point, there is not enough oxygen because you use a lot of energy. You don't get enough oxygen, so the cells have to do something else, and then apparently they switch to this, and then the byproduct of that is lactose, and that causes pain of the muscles. So under anoxic conditions or hypoxic conditions, our cells will also switch, I think. Yeah.
Hypoxic condition indirectly.
Yeah, that's right.
And the few people saying like the hypoxic condition is due to angiogenesis. Because
angiogenesis will bring blood to the tissue. So it cannot cause.
So the blood will carry more oxygen. So the other side can be a hypoxic condition.
I see.
Right? I mean,
with what I read, what I
it's
know
like
is
chicken
the
and
other
egg
way around.
problem.
That when there is hypoxia, hypoxia triggers secretion of VEGF and all these growth factors that invite blood vessels.
Okay.
Not the other way around. I mean, I think the first group who say what they say are right. I agree with those. Okay.
Thank you.
So let's try to finish. It's another strand of DNA from another chromosome. And during the replication, when the DNA opened, these two strands happen physically to be in proximity to each other. And comes an enzyme which cuts it and glues it wrongly. So you get a bit of DNA. There is a text running, which is the text which was on the blue chromosome, and then it continues in the red chromosome. It's like one of those mythological animals. The upper half is a woman and the lower half is a fish, or the upper half is a man and the lower half is a bull. Okay? So you end up, if this thing gets transcribed, and if this break happens inside the protein, and it gets transcribed, so this example that we looked at is the MLL gene. It's a gene on chromosome 11, and it has a partner gene, which is called AF4, which is on chromosome 4, two different chromosomes, blue and red. And what happened during the process of DNA replication, it was cut, and the head was from one animal and the tail from the other, and this is the fusion protein. It's an oncogene. It's a disaster, actually. It's implicated in 10% of all acute lymphoblastic leukemia cases and 80% of all infant leukemia. So it is really bad for a major factor for infant leukemia. So we went and did a study where we tried to, it's a transcription factor, where we tried to figure out which are the transcriptional targets of this oncogene. Okay, and that was published in the PNAS. Here is another way, and this will be my one before last slide, where a modification of the DNA can cause overexpression of a gene. So this is the situation. This is in a study that we did on leukemia in Down syndrome children. Turns out that children, you know what Down syndrome is? There is a trisomy of chromosome 21. So these children are 20-fold more likely to get childhood leukemia than normal kids. So the question is why? So we don't know why, but there are some differences that we already found. This is one of them. Here is a gene called CRLF2, cytokine receptor-like factor 2. It's a receptor. It's a growth factor, basically. In normal conditions, it expresses at some low levels. Here is a gene, P2RY8, which is involved in all kinds of cellular processes that need large amount of it. So this one is expressed at very high levels. It has a very active promoter. So what happens is that there is a deletion. All this bit of DNA from here to there is cut out and thrown away, and that it's glued back together. So when it's glued back together, what you get is this. You get the CRLF2 gene with the promoter of this guy. And therefore, this gene, which is a receptor, is expressed 20 to 100 times higher than in normal condition. So by deleting a bit of DNA, you place a receptor under the control of a very active promoter. So you get amplification, severe amplification of this receptor, which we found in these leukemic children. Okay. So I'll start here my next lecture telling you about DNA copy number changes, amplifications, and deletions. And I will sort of adiabatically go into the next talk on breast cancer. Okay, so thanks, I'll stop here. Yeah.
At what point in development did the translocation happen?
Good question. It's usually diagnosed when they are months old, between a few months and 2 years. So it's a very early stage. I don't think, I'm not sure what is the fraction, if there is any, of those that are born with the disease. I don't know, but it's a pretty early event in development.
So you told that cancer cells somehow is involving the fooling of the immune system. So what is that fooling you trying to explain? That means the fooling is downregulation of the immune system or upregulation of the immune system? Because I'm asking this question because the cytotoxic drugs, which are mostly used for cancer treatments, they also downregulate the immune system and as an overall process, because they can't differentiate between the targets and what is the non-targets. So then what is the gain of applying those medicines which are downregulate the immune system and somehow fooling those immune systems? So what is the gain of using those medicines in cancer?
First of all, I'm not a physician and I don't know the answer to the question, to this last question. And it's this business of gain. It's very delicate. Every drug, or nearly every drug, has side effects which are bad. So you have to walk a tight rope and balance things for the patient. Well, what's the benefit? So if the probability of the drug slowing down the tumorigenic process is 60%, and by affecting the immune system, you drive down the involvement of the immune system by so much. So you have to balance what is and sometimes you come up this way, and sometimes this way. I don't know. Now, what is the way that cancer cells do it? There are lots of ideas floating around. I don't think that there is one theory. I don't think that there is one accepted theory. And the frustrating thing about biology is, in many ways, and it comes back at you again and again, that there are some large-scale ideas and principles, like evolution, selection. But many of the things are different from one system to another. So if a breast cancer cell will evade the immune system differently, probably differently, than a colon cancer cell. They live in different environment, and the acidity is different, everything is different, and therefore they use different tricks. So I don't think there is a universal answer to your question. Okay? I'm sorry, I am not evading it. I don't really know the answer, a good answer. Yeah.
So you mentioned so many hallmarks and enabling factors and so on. Now
6 hallmarks.
6 hallmarks, yeah. And 4 enabling factors eventually, I suppose.
Yeah.
Right. Okay.
Yeah.
So now, when things start out, is it typically one of the hallmarks or something that happens, and it could it be any one of the different hallmarks, and then eventually all of the hallmarks or many of the hallmarks come into the picture? Which ones does it start out with, and which ones come in later?
I don't think that there is okay, I think the answer to the question has to do with every region on the genome has a different mutation probability or a different probability of deletions and so on. So those are hit faster than those where the probability is lower. It's a very trivial statement, but they are not organized by pathway. They are not organized by hallmark. And I don't think there is a logical sequence to follow. Okay? I think that the cancer cells that know how to, I would say that you get some advantage by proliferating. Because if you proliferate, then there is more replication. And if there is more replication, then there is more chance of adding damage to what you already have and to knock some other system out. So if the cell acquires the ability to proliferate and the ability to ignore the stopping signals, then it will start dividing, okay? Some of the cells will apoptose, but some will escape apoptosis. And so that's it. If you acquire the anti-apoptotic capability first, that will be less advantageous. Did I answer your question? So in that sense, logically, I would say that it makes sense to have the proliferation first and to knock out the apoptotic circuitry last.
Yes, so yeah, part of the question was related to the interaction between these different hallmarks and whether, I think you said, answered in the positive that having one will increase the probability of having the other or something like that.
Yes, right? In that sense, the mutation rate will go up if you divide more. Well, not the rate. The mutation rate is per division, per cell, per bit of DNA. So the mutation rate will not go up, but the number of events that are likely to get a mutation will be faster. Their rate will be faster. More divisions. So I think in that sense, that's the interaction. But that's it. I don't think that there is much interaction between evading between the genes that control apoptosis or some other process. Sometimes there are genes that play a role in more than one pathway, and if you knock that out, then that's faster, then there is some interaction. But normally, these things, they are viewed as independent events. Okay? Yeah.
This is a question on the Warburg effect. Could it be that the oxidative phosphorylation time scale comparison with glycolysis, with cancer, if getting some less amount of ATP but faster, via glycolysis is balanced with getting more ATPs but slower with oxidative phosphorylation, and given that cancer cells want to proliferate fast, maybe with lesser amount of ATP, is there a preference in that kind of optimization? I'm just wondering whether there's literature on that.
I don't know the answer, because I think the cells at some point reach some steady state. And so I think the rate of production is, I'm not sure, but I don't think that is an advantage. You have to get food in order to glycolyze. So you have to have a supply of nutrients, and from each glucose molecule, you produce, instead of 18 ATP molecules, you produce 1 or 2. I mean, it's not a good deal. So that I think they must gain elsewhere, and it's not clear where.
So a very naive question, what is the role of the clearance of the damaged cells or clearance of the cells in cancer or in apoptosis?
Clearance to get the debris out?
Exactly. So after a certain cycles which they're damaged cells, so it's called the hypersensitivity. So what is the role of the type 1, type 2, and type 3 hypersensitivities in case of cancers and apoptosis? That's why you want to know if you...
I'm not sure I understand the question. I'm sorry.
Okay, so after a certain time, some cells will be damaged. So there is a clearance mechanism of the body to clear out those cells.
The dead cells.
Yeah, the dead cells and the damaged cells. So in cancer, what is the role of this, the impaired mechanism of damaging cells clearing out?
I don't know that. I don't know. Dead cells, that's debris which is cleared away in the bloodstream or in the, and I think it's, I'm not sure. I don't know. I haven't read about the role of the... I have to stop this talk before I crash. So I really don't know. I have to think about it, but I didn't even think about it. On the organism level, what is the role of the clearance? I don't think that there is a business of a bit of the cancerous cell infecting a healthy cell, if that's what you have in mind.
No, so these things comes into my mind because in some cases, this hypersensitivity procedure, that means the clearance procedures, is hugely related to the immunological disorders and mainly the autoimmune problems. And in some studies, it is found that some kind of autoimmunity is just opposite of cancer, and some studies suggest that if you have autoimmunity in certain stage of life, then after 15 years or after a certain period of time, you will have a higher chance of cancers. So that's because I wanted to know it. So for me, it seems like that the immune system it's like a global attractor, and the other things as like a cancer is like a limit cycles, and it can't get out of this, and some oscillations and flare-ups in case of autoimmunity. So it's a just two sides of the same coin, or like that. I just wanted to know what to comment.
It's an interesting idea. I don't know. I don't have any intelligent thing to say about it. I worked on autoimmune diseases. I can tell you that they are very complicated. And whether there is any crosstalk, when you say that people know that if you have, I don't know, lupus, then your probability of getting cancer goes up or down. I don't know the fact, so I cannot, I don't feel like trying to philosophize about it, okay?
So let's thank
Okay.